Good morning, everybody. Welcome to day one on week 6 of the summer semester. This week is the last week of the semester, so we have finals by the end of the week. Make sure that you check online for the deadlines that we have for the upcoming assignments. Last week on Thursday, the last thing that we mentioned was the cleaning action of soaps. We learned how to synthesize soaps. And just as a reminder, a soap is made specifically by taking a triglyceride. It could be a fat or it could be an oil, and then we saponify it, meaning we are going to hydrolyze them under strong base conditions. Remember that the products of that reaction is going to be glycerol, which is the component of the alcohol, and a carboxylate salt. And the soap is the carboxylate salt. When it comes to a soap, remember that the soap is going to have two components, one that is polar and one that it is non-polar. The polar portion of your soap, meaning the ionic portion, is going to be represented by the circles that you see in the drawings here. The tail is going to be the non-polar side of your soap. As you can see here, when we are cleaning something, what happens is that the tail, because it's non-polar, and grease is also non-polar, they're going to be interacting with each other. Then after the molecules orient themselves, okay, with the correct interactions with water and the grease, we can see that through agitation, more molecules interact with that grease, okay? And in order to complete the cleaning of that surface, you can see that micelles, which are these structures that are going to have a polar head and the non-polar tails interacting with the grease, are going to solubilize that grease, and that is going to be removed from the surface that you're cleaning. Now, today we are going to move on to another lipid that also contains fatty acids. Now, phospholipids or glycerol phospholipids specifically, let's understand that these molecules are very important in biological systems, okay? Because when it comes to these compounds, understand that these are the most abundant lipids in cell membrane, and they play an important role in cell permeability. What cell permeability mentions is that this is the ability to let other molecules, compounds that need to get into the cell or get out of the cell. And understand that this is going to be what makes the plasma membrane. So that's why it's such an important structure. Now, in biochemistry, you may have noticed that we always have cartoons to represent these molecules, because when we look at them at the chemical level, these are large structures with a lot of atoms. So in biochemistry, in order to give a more simplistic view, but still meaningful, we utilize a lot of drawings. So when it comes to phospholipids, we can actually observe that the phospholipid is actually made from glycerol, 2 fatty acids, a phosphate group, and an amino alcohol. So I'm going to label all of those components in the molecule that we have below. So the glycerol portion, I'm just going to zoom in to this area of the molecule so we can have a better view of each of the components. So this, which is the glycerol, we could see it in this portion of the molecule. This is where the glycerol is. Okay. Now, the fatty acids, so you can see we have 2. One right here, one right here. Notice that the fatty acids connect to the glycerol backbone, and we call it backbone in biochemistry because it is holding. It's kind of like the spine. That's why we call it a backbone. So when it comes to this, let's notice that when it comes to functional group, there are ester functionalities connecting the fatty acids to the glycerol in a glycerol phospholipid. Then you guys can observe that we have a phosphate group. In organic chemistry, phosphates are not as common as in biochemistry. This is a functional group that is very important in biochemistry. And in addition, it is part of the communication system of the cell. But we are not going to be dealing with cell signaling, but it's an interesting topic. Lastly, we have an amino alcohol. And as you can see, that amino alcohol is right here, where the O from the alcohol was what bonded to the phosphate here. And then you can see that the amino is a quaternary amine that we have here, okay? So that's how you identify the different components. Now, when we look at this molecule overall, let's understand that phospholipids, in a way, the drawing is going to be a little bit similar to specifically the soaps, but this is the difference between them. When we are drawing a phospholipid, we have a polar head like we saw on the soap. And you can see here that the polar head is going to basically be defined by the amino alcohol and the phosphate, okay? So that's where you see that green box. Then the hydrocarbon tails, as you can see, there's two of them, then are going to be squiggly lines. If you don't see the polar head as a rectangle, sometimes we see it as a circle. So understand that another drawing, and remember that this is my little symbol for redraw, is just drawing that polar head and then putting two squiggly lines down, okay? Remember that for a soap, we only have one squiggly line, specifically because we only have one nonpolar tail. Let me just, so this is the polar head. This is just another way that this can be illustrated, okay? But as I mentioned, when it comes to phospholipids, they're very important molecules. As I mentioned, they are going to be found in cell membranes, but it is not the only component of a cell membrane, okay? When we look at an animal cell, and you can see the little drawing here, remember that that cell membrane is going to be the outer layer in your cell. So it's going to be that green area. That is what's going to keep the stuff from inside the cell protected from what is outside the cell. And it's also going to mediate the things that come in and out of the cell. But when we look closely at the cell membrane, let's observe all the different components that are present in the cell membrane. As you can see here, we do have a phospholipid. Now, this phospholipid is what we know as a phospholipid bilayer. Why is it called a bilayer? Because you can see that we have two layers of it. When it comes to looking at the structure of the phospholipid bilayer, we can see that this is the polar head, this is the tails, and in the other layer, the tails are interacting with the tails of the first, because remember that those areas are non-polar, so that is going to be interacting with each other, okay? That's why the areas where we see the tails specifically, as you can see how it's been pointed here, this area is known as the hydrophobic region. But it makes sense. They are non-polar. They're going to be afraid of water. So that's why they're called the hydrophobic region. Now, when it comes to the phospholipid bilayer, we also have a hydrophilic region. And when it comes to this hydrophilic region, notice that that is going to be the polar heads. But again, that makes sense. That area of the molecule is going to be polar, so it can interact with water. That's why it's hydrophilic, in love with water. Now, notice that in addition to that, what we have in terms of other molecules, and I haven't introduced them, is cholesterol. And we're going to see later how cholesterol is a steroid, and it is part of the cell membrane. We also have proteins. And in terms of protein, we're going to talk about them in more detail in chapter 16. Notice that we also have carbohydrates. So understand that the cell membrane has a variety of components in it, and they're all important, and they all play a role into how we are going to mediate things that are happening at the cell membrane. Now, there is a specific model that it is utilized to understand this variety and complexity that we have in the cell membrane. And that is known as the fluid mosaic model for cell membranes. As you can see, the fluid mosaic model explains the structural components of biological membranes, okay? Now, let's understand that the word mosaic refers specifically to the different components that we have in the cell membrane. The proteins, the carbohydrates, the cholesterol, the phospholipid bilayer, so on and so forth. Now, one of the important things that cell membranes are not rigid structures, okay? They are more fluid, okay? So understand that in this fluidity is because specifically when it comes to the structural components of those fatty acids that make that phospholipid that is there, unsaturated fatty acids are the majority, okay, but not all, but they are the majority of the fatty acids that we see in there. We learned previously in this chapter that whenever you have things that are unsaturated, because they have that carbon-carbon double bond, one or more, remember that that is going to make the structure have what we call in biochemistry a kink, and that is going to make it more fluid-like, because technically things that have more unsaturation in them are going to be more liquid. But again, it's not going to be completely liquid, because if not, it wouldn't have a correct consistency, okay? Now, let's understand that cholesterol also plays an important role in the structure of that fluid mosaic. And cholesterol as a molecule, actually, because of the dispersion forces that can occur with the nonpolar tails of the phospholipid bilayer, then that actually means that we are going to see some rigidity in the membrane. So that's why it's not completely liquid, it's more of a fluid. And people actually have done studies to understand how inside of the cell and many components of the cell are more gel-like, okay? So it's not completely a liquid. It looks a little bit like a solid. Now, or it has the consistency as a solid. It's hard to define. Let's just call it, it is more like a gel. Now, all these components, the proteins, the carbohydrates, serve a very important purpose in the cell membrane. And that is because cells need to communicate. They don't have an actual verbal language, but they have a biochemical language. And that biochemical language, it's going to allow these chemical compounds to be interacting with one another via these proteins. okay, that are going to be in the lipid bilayer. Now, let's now talk about how things are actually transported into the cell and outside of the cell. So in order to talk about that, we're going to learn the different forms of transport that happen at cell membranes, okay? Now, when it comes to this, let's understand that there are 3 different forms of transport that we're going to talk about in this class. The first one is diffusion, okay? And diffusion is also known as passive transport, okay? So diffusion, as you can see in the first image here, is going to let things from outside of the cell to come inside of the cell, and things are going to move from higher concentration to lower concentration, okay? Now, not all molecules have the ability to do this. There are a number of molecules that are known to be able to do this, because if you think about it chemically, this molecule needs to have a specific characteristics to actually pass through the polarity that that lipid bilayer has. So it is known that the molecules that can actually diffuse include small, non-polar molecules like O2 and CO2. They're small, they're non-polar, and they're able to do this. There is a special polar molecule that can also diffuse into the cell. And I think I haven't found any other examples other than this small polar molecule that can actually undergo diffusion. The other molecule that can undergo diffusion is going to be one of the most important compounds that we know to date, water. Water is polar, but as I've been telling you guys throughout the semester, water is very special. Water doesn't behave like other molecules that share the, similar atoms or similar molecular weight. Water is going to be one of those molecules that, even though it's polar, okay, and to date, I haven't found other examples, but water can actually get into the cell through diffusion, okay? Now, the other form of transport that we have is called facilitated transport. Understand that facilitated transport is also a form of passive transport. It's just that in the book, they didn't define it that way. But diffusion and facilitated transport, both are passive transport, okay? Now, what is the difference between facilitated transport and diffusion? Actually is the use of a membrane protein, okay? So in facilitated transport, we are going to have a specific tunnel that is going to allow our molecules to enter the cell, okay? And because that tunnel is going to be specific for the molecule that we're interested in, it actually increases the rate of diffusion. One of the biomolecules that we already talked about in chapter 13 that can actually get into the cell through tunnels is going to be glucose. And other sugars have the same idea. These molecules are polar. And in the cell, there's actually proteins that are dedicated to be tunnels for different sugars, okay? But glucose has its own like VIP entrance tunnel to get into the cell. The last type of transport that we have here is active transport. Now, active transport is going to move ions against a concentration gradient. What ions actually are able to do this? Potassium, calcium, sodium. These are important ions inside of the cell, okay? Notice that whenever we have an active transport, we are also going to have a membrane protein that is going to create that tunnel for having these ions, okay, come inside of the cell, in and out of the cell. Now, one of the important things to note is that out of these three types of transport, in active transport, that is the only one that needs energy in order to transport these ions. And remember that even though we're not going to talk about it in detail, energy in the cell comes in the form of ATP. So you need the hydrolysis of ATP, and that energy that is released specifically from that event, from the stored energy on the bonds, is what allows these ions to be transported through active transport. Any questions on membranes or transport? Then let's move on to the next concept, which is sphingolipids, okay? So when it comes to sphingolipids, this is going to be the last member of the lipid family that contains fatty acids, okay? So the other members of the lipid family that contain fatty acids, okay, including waxes, triglycerides, phospholipids, understand that sphingolipids are special because they are not connected to glycerol. And out of the three that I just mentioned, remember that waxes do not have glycerol. They have an ester bond, but they don't have glycerol. It's only the triglycerides and the triacylglycerols, triglycerides and phospholipids that have glycerol as a backbone. In sphingolipids, specifically, we have the fatty acid connected to what is called a sphingosine, okay? So the sphingosine is what's going to connect the fatty acids, okay, for this molecule. Now, once we have connected a fatty acid to the sphingosine, and notice that when we look at this at the chemical level, okay, the fatty acid connects specifically to the amino portion of the sphingosine. So let me label this in a moment. So we are connecting our fatty acid to the amino portion of the sphingosine. As we learn in chapter 14, when an amine and a carboxylic acid are linked together, understand that what we form is an amide bond. So in a sphingolipid, it is the only one out of our lipids that we have discussed so far that a fatty acid is bonded to the bigger molecule through an amide bond. What we've seen so far is ester bonds. Now, understand that this ceramide, which is what it's called, whenever you have a sphingosine linked to a fatty acid, that is what what it's called. That is a ceramide, okay? When that ceramide, which as you can see is found in high concentrations in the cell membrane, is then bonded to a phosphate group in an amino alcohol, that's what makes other type of lipids that are known as sphingomyelins, okay? And sphingomyelins specifically are going to be abundant in white matter, okay? And is going to be the main component of the myelin sheath, which is this tissue that is surrounding nerve cells, and is what actually insulates and protects the nerve cell, and it actually helps with the speed of the nerve impulses, okay? So understand that when it comes to sphingolipids, they're going to contain a sphingosine. That sphingosine once is linked to a fatty acid that is known as a ceramide. And then if that ceramide is bonded to phosphate and is bonded to, for example, choline, which choline is the amino alcohol, then that becomes a sphingomyelin, okay? And this concludes our topic of the lipids that have fatty acids as part of their structure. Now we are going to move on to talk about the other family of compounds that are also considered lipids, and those are steroids, okay? So let's look at steroids, how they look structurally. So what is a steroid? A steroid is going to be a molecule that it is very important in biology, in chemistry, and in medicine. What makes a molecule a steroid is having in its structure a steroid nucleus. And what is a steroid nucleus? It is a continuous chain, okay, of cyclic forms. And you can see here that the steroid nucleus needs to be in that arrangement. A 6-member ring bonded to another 6-member ring, and then we have another 6-member ring and a 5-member ring, but they need to be attached in that manner, okay? Now, steroids are very important because, for example, the best known and most abundant steroid that we have in the body, cholesterol, is an important feature of cell membranes. But understand that steroids also have other functions, okay? They are going to be the reactants to make sex hormones, to make adrenal hormones, to make bile acids, and bile acids are, released from the gallbladder for us to deal with fats. And they're utilized to make even vitamins like vitamin D, okay? Cholesterol is the main one that is utilized to synthesize many of these, okay? So let's now focus on, again, the most abundant and best known steroid in the body, which is cholesterol. Now, where do we get cholesterol? And you can see the structure of cholesterol right here. Cholesterol is obtained from meat, milk, and eggs, okay? That's how we obtain it from our diets. Now, if you are a vegan, can you make cholesterol? Of course, because cholesterol, it is actually synthesized in the liver, okay? So again, as humans, we are technically animals, so we are going to have the ability to synthesize this important molecule. In the liver is where it is synthesized, okay? Now, why is it that we need cholesterol, as in the body? We need it in the cell membranes because, as I mentioned to you guys, you cannot have a membrane that it is fully unsaturated fatty acid because it's going to make it liquid. Having the cholesterol in that membrane is going to be given that membrane more like a gel-like composition, okay? It's more fluid, it's not completely liquid. Now, we need cholesterol, okay, for the brain. We need cholesterol for nerve tissues. We need it because it is important for the synthesis of steroid hormones, and it is needed also for synthesizing important fat-soluble vitamins like vitamin D. Now, cholesterol, it is something that we need to watch out because you cannot have, very, very high levels of cholesterol, okay? because understand that if cholesterol, you have them in your body in high levels, it can actually form a plaque and clog arteries, okay? Now, cholesterol has a specific transportation system in the body, and that is known as a lipoprotein, okay? So lipoproteins is what carries cholesterol, okay, in the body. Also, it carries triacylglycerols. So the triglycerides are also carried in these molecules. And you can see the structure of a lipoprotein right here. Let me just zoom in. So in a lipoprotein, as you can see here, is called a lipoprotein because what the outside of the lipoprotein is going to be phospholipid. The word protein is because there are these structures, and we're going to learn about them in chapter 16, that are made out of amino acids that are also on the surface of the lipoprotein. But look, inside of the lipoprotein, what we have is triglycerides and cholesterol. So understand that lipoproteins is what we use to carry, okay, cholesterol in the body. Now, not all lipoproteins are going to be the same. So when we look at the image that we have here, okay, notice that 25% of cholesterol comes from foods that you eat, okay? 75% of the cholesterol in your body is made by your liver. So that's why if you consume a vegan diet, you're still going to have cholesterol in your body because it is made in the body, okay? Now, it is unfortunate to see that 33.5% of American adults actually have high cholesterol, and it is a concern. Just like I said, because this may cause, you know, clogging your arteries, and that may lead into problems with the heart, problems with circulation. So that's why it is important to know. Now, when it comes to the lipoproteins, as I was mentioning, understand that there's going to be two types of lipoproteins that continuously, and they can check the levels when you get blood work done, okay? We have what are called HDLs, or in other words, high density lipoproteins, okay? We also have LDLs, which are known as low density lipoproteins. And you can see in the box that looks like a chart, what are the healthy levels for LDLs and HDLs, okay? Also, you see the levels for triglycerides and total cholesterol in the body. And all of that is done through blood work, okay? And you can just get the lipid profile test. Now, when it comes to the lipoproteins, the high density lipoproteins, it is known as good cholesterol. And the reason why it's known as good cholesterol is because that's the type of lipoprotein that is going to transport cholesterol to your liver, okay? And it's going to prevent the buildup of the cholesterol in your arteries, okay? HDLs also remove cholesterols from your bloodstream. That's why when you're looking at your levels, you want to have a good level of HDLs. Now, LDLs, you do not want to be out of the normal range, because as you can see, LDLs are known as bad cholesterol, because LDLs are what's going to transport cholesterol from liver to heart, for example, or to muscle. But their role is only to carry and dump it there. So if you have too high of LDLs, then because they are going to be depositing cholesterol, and if your HDL is not keeping up with the dumping of the LDLs of the cholesterol, again, in muscle and heart, that's where the problems come from. And having high LDLs result in arterial plaque buildup, in other words, clogging, okay? And this may lead to a heart attack or even a stroke. That it is important that, you know, when you talk to your doctor, if your lipid panel is taken and it is out of range, that you talk to your doctor and make sure that it is on the correct range, okay? Now, let's talk about other molecules for which cholesterol is also important, okay? Let's understand that when it comes to other biomolecules, okay, that are synthesized from cholesterol, these are going to be their structures, okay? Hormones are going to be defined, as you can see, as a product of living cells that are going to circulate in body fluids, okay, and produces a specific stimulatory effect. Basically, these are going to be hormones. Not all hormones in the body are steroids. Let me just specify that. Sex hormones and adrenocortical steroids are going to be specific biomolecules that are utilized in signaling at a specific point during development, okay? And there's other ones that are triggered depending on, you know, your health or what is happening environmentally. Now, when it comes to the hormones that are steroid-based, you can see here that we have estradiol, testosterone, and progesterone. All of them are sex hormones. They are released in the body during puberty in order to signal the body that now our body is going to be maturing, and then developmental changes happen to the body. All of them are considered steroid hormones. Again, they're synthesized from cholesterol, and you can even see the steroid nucleus in each one of them. Adrenal cortical steroids, as you can see here, we have aldosterone and cortisol. There are going to be other signaling molecules that we have, and specific, they are going to be released by the adrenal gland, which is this tissue that is on top of the kidney. And they're going to be, again, utilized as signaling molecules in the body. Cortisol is one of the ones that it is checked sometimes by blood, because that is the molecule that it is involved in the stress response. So if you live with biochemical stress, just because environmental issues are affecting you, you have inflammation in your body, cortisol may be one of the molecules that your doctor may check to just see if you are living with stress, okay? But again, it is important to know that when it comes to these molecules, they're synthesized from cholesterol. Now, in the body, we also have vitamins that can be synthesized from cholesterol. And specifically, vitamin D is the one that it is synthesized from cholesterol. What are vitamins? These are going to be organic compounds that are required in small quantities for normal metabolism, okay? Understand that the fat-soluble vitamins are actually also lipids, okay? And they must be obtained from diet, specifically A, E, and K. D, it is one of the ones that can be synthesized in the body from cholesterol specifically. Now, this is the importance of fat-soluble vitamins. And understand that fat-soluble vitamins are actually stored in the fat tissue in our bodies. I wanted to give you guys at least also the structure so you see how these fat-soluble vitamins have a lot of hydrocarbon components to them, and that's why they're mostly non-polar. So let's first explore vitamin A. Vitamin A is important in vision, reproduction, bone health, in the immune system, also in skin. And you can look at the structure, which is the first structure that we have in the bottom left. Then we have vitamin D. Vitamin D, very important for, again, bone, calcium absorption, the immune system. Vitamin D is the only one out of these that are going to be synthesized from cholesterol. You cannot see in a way specifically the steroid nucleus in it, but we can see in the middle of the molecule that 6-member ring and that 5-member ring. But understand that that one is synthesized from cholesterol. And vitamin D, actually, you can consume it, but it is important that when you're outside, please use sunblock, but you need the sun, okay? I can talk about from experience after, working at home during the pandemic. Vitamin D is one of the things that, at least personally, I was deficient in. When I went and did my annual checkup, my doctor was like, you need to take vitamin D. You haven't been outside much. So I take supplements to increase the levels of vitamin D. Then we have vitamin E, and you can see that is is very important for the immune system, and it helps with flushing toxins. And lastly, we have vitamin K, which is important for blood clotting and bone health. You also see here different food items that are going to be particularly important in these vitamins. But when it comes to this, I'm just trying to illustrate even the idea that vitamins that are important in the body are also known as lipids. Any questions when it comes to steroids, cholesterol, hormones, or vitamins? I really don't have a specific answer because I'm not a medical professional. How do we increase the HDLs? But I can think of ways to increase them through diet. I imagine that that would be the recommended. I know for people that have, but that's more for the triglycerides. I don't know if there's, I imagine there will be medication for biochemically controlling the LDLs in the body, but I imagine possibly diet will be the suggestion from doctors. I don't know the specifics. Any other questions? Then let's move on and start talking about chapter 16, which now we are going to understand amino acids, proteins, and enzymes. So first, let's define what is a protein, because you guys have heard me in chapter 15 talking about proteins. So when it comes to biomolecules and in biochemistry specifically, we look at the whole area of biochemistry in terms of carbohydrates, which we already talked about in chapter 13. We think about them in terms of lipids, which we just finished talking about it in chapter 15. Proteins and amino acids, which is what we are going to be dealing now, and in the next chapter, nucleic acids. That's basically going to be the big 4 families of compounds in biochemistry. Now, proteins are going to be polymers, and remember that the word polymer defines that these are going to be long chains of a basic unit. The basic unit for a protein is going to be amino acids, okay? Now, even though proteins are made from the same 20 different amino acids, okay, they are going to have different characteristics and functions depending on the order of those amino acids that make that protein, okay? So it's just the different combinations of the amino acids that actually give rise to different proteins. Now, proteins have different functions, and we're going to see how these different functions can be categorized, but some of them include structure. Sometimes they are going to be regulating biochemical reactions via enzymes, And sometimes protein can even transport or store important molecules. As you can see in the last bullet point, we have hemoglobin and myoglobin, which are the molecules important in transporting oxygen in the blood. So understand that the main thing here is that you understand that the amino acid is the basic unit of a protein, and there's 20 of them. There's actually more than 20. It's just that in most animals, or most organisms, I should say, they share the same 20. There's some specialized amino acids that are in certain parts of the body, but it's not among organisms. So that's why we always teach the basic 20. Okay, now let's look at amino acids at the chemical level more closely. So when it comes to the amino acids, the reason why we call this basic unit an amino acid, it is because of the functional groups that are present in it. When we look at an amino acid, understand that all amino acids are going to have an amine group that it is protonated. So that's why we call it the ammonium group. They're going to have an alpha carbon, and that alpha carbon is going to have an alpha hydrogen. Every amino acid has a carboxylate group, which comes from a carboxylic acid, but because of the pH of the blood, that's why the amino group gets protonated and the carboxylic acid one gets deprotonated, because remember that the pH of our blood on average is 7.4. Now, every single amino acid also have what is called an R group. And remember that R group, that is a concept that I introduced to you guys in chapter 11. R means rest of molecule. And this R group, which is going to be the side chain for every single amino acid, is what makes them different. So understand that every single amino acid is going to have a different R group. Now, when it comes to the amino acids, understand that the amino acids can be grouped in 4 different families, okay? The first family of amino acids is going to be called the non-polar chain amino acids. And you can see them right here. The side chains is what you see in the color boxes. That is what the R groups are, okay? Now, if we go back into what we have learned so far in the area of organic chemistry, let's now define what are the main functional groups that we see in the side chains for non-polar amino acids, okay? So when we look at the family of compounds and functional groups in the side chain of non-polar amino acids, what do we see? We see methyl and things that have carbons and hydrogens. What are they called? There's benzene, great. So remember, carbons and hydrogens, benzene. What is the big family that we call them? There's not a thiol. I will talk about methionine in a moment. So we see a lot of carbons and hydrogens. What do we call those? Organic functional groups that have carbon and hydrogens. What are those called? Excellent, hydrocarbons. And look at the hydrocarbons. We have CH3, we have isopropyl. This is isobutyl, this is sec-butyl. Here we have a benzyl group. Here we have a cyclic compound. Okay. Out of all of these guys, glycine is the only one that has a hydrogen as a side chain. Okay. Because of that, glycine is the only amino acid that is not chiral, because the carbon atom has two of the same group. The other carbon atom in that amino acid is a trigonal planar. So actually, it cannot be considered chiral, okay? Now, there's one functional group here that is different than we haven't seen before, and that's the one in methionine. This group, you guys, or the side chain, for methionine, it is a functional group that we haven't seen before. This is known as a thioether. Because remember that we learned before that when an oxygen atom has carbon chains on either side, that is known as an ether. So here, because we have a sulfur instead of an oxygen, that's why this is known as a thioether. Okay, so that is methionine. But understand that the majority of the nonpolar side chains are going to be hydrocarbons, hydrogen, which that's going to be glycine. And then we have the thioether. Okay, that's what mainly we see here. Now understand that when it comes to the amino acids, we're going to have a full name for them. So you can see glycine. And then we're going to have a one-letter code that is given by a single letter, G. And then we're going to have a three-letter code, the GLY. That's the three-letter code for the amino acids. And every single amino acid has one, okay? Let's continue exploring the different amino acids. Let's now look at the amino acids that are known as polar side chains. As you can see, the polar side chains are going to be given by that lavender color, okay? So if we go back into our organic chemistry library, what functional groups do we see in the side chains of polar amino acids? Let's put it in the chat. What functional groups do we see here? Great, yes, there's alcohols. You can see serine, threonine, tyrosine have an alcohol. What are the functional groups we see here? That's not an amine, close, but that's not an amine because that nitrogen is bonded to a carbonyl. I don't see any aldehydes. I'm looking at the areas that are in the lavender color. There's no aldehydes. What about cysteine? The letter C, what is that functional group? Great, that's a thiol. What is the functional group that we see in asparagine and glutamine? Great, that's an amide. Okay, again, I'm talking about the side chains. So again, the second family is going to be the ones that have a polar side chains, and we can see the members there contain alcohols, thiols, and amides. Then out of the electronically charged side chains, we have the acidic ones. So when we look at aspartate and glutamate, okay, that side chain used to have a hydrogen on the oxygen. Again, the reason why we don't see the hydrogen on the oxygen is because at pH 7.4, that hydrogen doesn't live there. So what is the functional group if that oxygen has a proton that that side chain will be. So if you're considered acidic, okay, what is the functional group in that side chain? We just talked about them in chapter 14. Great, they used to be carboxylic acids. Again, these carboxylic acids at the pH of the blood lose the proton. So that's why you see them as O-. So if you have an acidic amino acid, that's because the side chain are carboxylic acids. Right now they look as carboxylates, but we learned that carboxylates come from carboxylic acid. Now, when we look at the basic amino acids, what functional group do we see? At least in lysine and histidine. What is the functional group in basic amino acids? They're basically weak bases. What are the functional groups that we see there, at least in lysine and histidine? Great, those are amines. Lysine is just a primary amine. Histidine has a heterocyclic amine in it. Arginine as an amino acid has a special group that we haven't seen before. So this area of the molecule, that is known as a guanidinium group. And guanidinium groups in organic chemistry is not a functional group that is very common, okay? Just understand that this nitrogen is the one that is going to be the most basic out of the whole system. And that's the one that releases protons or grabs onto protons, okay? But these are the main families, okay, of amino acids that we have. Any questions about the amino acids? Now, one more thing before we go on break, because I know that we're going to utilize it. So what you guys have on the slides is actually the condensed structures for the amino acids. Structures. I'm going to try to find, oh, right here. So in the chat, let me see if it opens. There you go. So in the chat, I put in the link for a list of amino acids that have the amino acids in skeletal structure, okay? One of the things that we're going to learn today is how to define the amino acids present in a peptide. And I want you guys to also have that image because the computer program that I utilize for generating my peptides gives you the amino acids in skeletal structure, okay? Let me see, there was a question. So glycine is the one that has the hydrogen side chain. I'll go back into the slides. I just wanted to make sure. Oh, for histidine. Give me one moment. Let me just go back. So for histidine, this nitrogen is the one that is going to specifically donate the proton or grab onto protons. And for lysine, this nitrogen, because it's a primary. Did I answer your question, Gia? I don't know if I did.
Oh, it's just a different structure. Like I've never seen the little arrow or looking thing. I don't know if that was like a different.
No, think about it. This is just a heterocyclic amine.
Oh, gotcha. Okay.
So the nitrogens are inside of the pentagon.
Gotcha. Okay, that makes more sense. Okay, thank you.
No problem. Kayla, does this answer your question, the guanidinium group? Just let me know in the chat. No problem. Any other questions about amino acids? Then let's go on break.